Welcome everybody to the first video in the Intro to Microbiology series. And this video is going to talk all about the different type of microbes that we are going to be discussing this semester. So in this class, which is microbiology, we are going to be studying things that are too small to be seen with the naked eye. Now that definition is really a kind of general, but it is for a reason. You'll notice I used the word things, and please know I'm not talking about dust particles or anything like that. We are mainly going to focus our attention on microscopic pathogens. And pathogens are things that can make us sick. Now, many of these small things that I'm referring to are going to be microorganisms, which by definition, micro means very small, and then organism is a living thing. So many of the pathogens we're going to be talking about are living organisms, okay? But two of the microbes that we talk about are not technically considered to be alive. So I'm normally going to use the term microbe rather than microorganism. So when I use the term microbe, I am referring to those microscopic pathogens, whether they be living or non-living. And we'll talk more about that here in just a minute. So we're going to talk about 8 different groups of microbes. 6 are considered to be living and 2 are considered to be non-living. So let's go ahead and get into the details. So I apologize, I forgot to animate one of the things for viruses, so we're going to ignore that for now. So the first one that we are going to talk about, the first microorganism, are bacteria. And these are prokaryotes. Okay, now you're going to hear a lot of terminology on this slide that you're going to actually talk about in depth in future Unit 1 videos. Okay, so I'm going to give you just a very brief overview, but I'm just kind of trying to introduce some of these terms now. So prokaryotes are unicellular organisms. Uni meaning one. The entire organism is one single cell, okay? And the prokaryotes are much smaller and structurally simple than eukaryotes, which we have eukaryotic cells, okay? So in other words, bacteria are made up of a single organism that lacks a single cell that lacks internal structure. It is very structurally simple. Bacteria are also pathogens. Some of them are pathogens, not all, but some of them. Remember, pathogens means that they can make us sick. So there are some bacteria that do cause disease, but not all of them. Some bacteria are actually very helpful. They make up what's called our normal microbiota. That's going to be all the bacteria that is in our body, maybe helping with things like digestion, but also the bacteria that are normally part of, like on our body, like on our skin, for instance. And we're going to talk a lot about our normal microbiota and pathogenic bacteria in this course. We tend to focus more on bacteria because that's what we come into contact with on a very regular basis. Bacteria divide or replicate through a process called binary fission. So essentially a bacterial cell will kind of stretch outwards and eventually pinch off into 2 cells. And that's how the bacterial populations increase in numbers. Okay, it's a form of asexual reproduction. And we're going to talk more about that in Unit 2. Bacteria, when we get to the lab portion of class, we're going to be talking a lot about the shape of bacteria. And there are 3 common shapes. That's coccus, which are spherical cells, bacillus, which are more rod-shaped cells, and spirillum, which are more spiral-shaped cells. So again, right now I'm just trying to introduce you to a lot of the terms that you're going to hear when we talk about bacteria. Bacteria also have a cell wall, which is going to help the cell maintain its structure. And the main component of the bacterial cell wall is peptidoglycan. We'll talk a lot more about that later in this unit. All right, on to the Archaea. The Archaea is the other group of prokaryotic organisms. And so again, that means they're structurally simple. They are also unicellular. Okay, so what you see here, this is one single cell, and that is the entire organism. Archaea are found in extreme conditions, areas that are really hot or super salty. So really, they exist in places that we normally do not encounter. So the Archaea don't really ever affect us, okay? So we're not really worried about getting sick from the Archaea, okay? All right, so bacteria and Archaea are the only two groups that are prokaryotic. Okay, so we're now going to move into some eukaryotes. These are going to be larger cells that have a lot of internal structure. So fungi are the first, so they're eukaryotic. Some fungal species are unicellular, some are multicellular. There's a lot of diversity within the fungi. A lot of them are saprophytes, which is just a fancy word for decomposers. So they break down dead and decaying organic matter, and that really helps with recycling nutrients back into the earth. They also have a cell wall that is made up of chitin. Next up are algae. Algae are a type of protists. We will talk about the fungi, algae, and protozoans in Unit 5. That's what we end the semester with. So algae are eukaryotic. They are also unicellular or multicellular, depends on the species. People often confuse them for plants because they do have a cell wall made up of cellulose, much like plants, and also because they are capable of photosynthesis. And so algae look like plants, but they are not. They don't have like a root structure or anything like that. All right, for the protozoans, they are eukaryotic. These are all going to be unicellular. They do not have a cell wall, which is pretty unique, and they are very motile. They move around a lot. So when we get to Unit 5, we're going to kind of divide our protozoans based on how they move around. All right, our helminths. These are eukaryotes, and helminth is really a fancy science word for worm. Now, of course, we can see worms with our eyeball. We don't need a microscope to see them, but we include them in this course because they're part of their life cycle. They are microscopic. The eggs, the larvae, those are often microscopic, and they can make us sick, right? A lot of worms are, that we'll talk about, are parasites. But we will talk about them in Unit 5 as well. So they are eukaryotes, they are multicellular, and they are parasitic. Okay, and again, helminth is just a fancy word for worm. All right, on to viruses. So viruses are considered non-living, and we're going to have a huge discussion on them later in Unit 1. But they are not considered to be alive. So the subtitles that you're seeing on the screen are going to be covering up some of these words, but they are all on your PowerPoint. So viruses, like I said, are considered to not be alive. So we do not describe them as eukaryotic or prokaryotic. Okay, we also don't reference unicellular or multicellular, and that's because viruses are not made up of cells, hence why they're not considered to be alive. So we can't use any words that reference living things, okay? So prokaryote, eukaryote, those are words to describe living things. Viruses are not alive, so you won't ever hear me refer to viruses as either of those terms. So viruses are obligate intracellular parasites. Obligate means they are completely dependent upon their host to replicate. To make more viruses, a virus has to be inside of a living host. If you just have a virus sitting on your tabletop, it will not be able to do a single thing, okay? It cannot do anything unless it's inside a living host. That's the intracellular part as well. Intra means within. So a virus has to get into specific cells within that host. And then viruses are parasites because they harm the host, right? Nobody ever excited to have a viral infection. All right, as I already mentioned, they are not made up of cells. So the science term for that is acellular. When you see A as a prefix, that means without. So acellular means without cells. So again, that's one of the reasons they are considered non-living. All right, and that brings us to our final group, which are prions. Prions are also considered non-living. They are actually just proteins that have essentially gone rogue. We're going to talk a lot about proteins because they are the workers of the cell. We have tons of different types of proteins, and each of them have their own unique shape. And that shape allows them to do their job. So much like our hand, right? The structure of our hand allows us to pick things up. We can't pick things up with our nose, just like we can't breathe out of our hand. That's because the structure of these things dictates their function. So the shape of a protein dictates what it can and cannot do. So normally proteins are always one shape, forever and ever, amen. But with prions, for some reason, we don't really know what the trigger is. Some proteins will change shapes. This makes them infectious because this newly misfolded protein can interact with other proteins and cause them to misfold. Okay, and then eventually all these misfolded rogue proteins will kind of aggregate together, and they tend to kind of get together and form holes in brain tissue. Okay, and so if you look at the picture under prions, number 8, that's a picture of brain tissue, which is normally much more solid. All those little holes in it are caused by prions. So because prions are just rogue proteins, they don't have any genetic material, they don't have any carbs, it's just a protein, they are also considered non-living. Okay. All right, so they are mysterious because we don't know a lot about them. Prions are a hot area of research right now because they're starting to be linked to things like dementia and Alzheimer's because of their effect on the brain. Again, they make the brain tissue look like a sponge. So we call these spongiform diseases. So with prions, there's going to be a little bit that we talk about in this class, but there's so many more questions than answers out in the scientific community right now that we just kind of say they're a mystery. Okay, they're really cool, but they're a mystery. So what I want to wrap up this video with real quick is going to be, oops, my mouse is super sensitive. Sorry, hold on one second. There we go, is how we name microbes. So this is going to be for how we name living things. So this is not going to be true for viruses or prions because they are non-living. But everything else that we just talked about, groups 1 through 6, are named according to this binomial nomenclature. Binomial means there are 2 parts to an organism's name, okay? And so if we can go ahead and look down at the first example, Escherichia coli, with any living thing, you're going to see 2 words like that, normally pretty crazy looking. But the first word, which is always capitalized, that's the name of the genus. And then the second word, which is always lowercase, is going to be the species. Okay, so remember, there are going to be multiple species that fit within a given genus. Okay, so kind of think of it like the genus might be like canines, right? Dogs. But the species is going to be the different types of dogs that exist. Dobermans have a different species name, right? German Shepherds have a different species name, so on and so forth. So the genus is kind of more encompassing. The species tell us exactly which type of organism we're looking at. So you will always write the genus and the species. And again, always capitalize the genus, okay? Now, how we let people know if we're writing a report or something that we are referring to a living thing is by either underlining or italicizing the name, both parts of the name. So if you're typing out the name of the organism, you have a choice. You can underline it or italicize it. But if you are handwriting, like on an assignment or something for me, you always underline it. don't try to like write slanty or anything like that, okay? If you're handwriting the name of an organism, underline both the genus and the species. But again, if you're typing it, you have an option. So you can see that here under the example for formatting rules. You can just underline it or you can just italicize it, but please don't do both. Lastly, if you are going to be writing a report and you are going to have to reference E. coli over and over again, just like with anytime you're going to make an abbreviation in a document, you always spell it out correctly in its entirety the first time. You put the shorthand in parentheses, and then from then on, you just abbreviate. And so I know the captions here are kind of covering the example, so please look at your PowerPoint. But if we were studying Staphylococcus aureus, and again, I can't read the exact words on the screen. If we were saying, you know, one bacteria we will study in lab is Staphylococcus aureus. Notice I said the full name, and again, you would either be underlining or italicizing it. And then right after, in parentheses, you would put capital S period aureus. Okay, so you're really just abbreviating that genus name. Again, it should still be italicized or underlined. You would close the parenthesis, put the period, end of the sentence. From then on, you just say S. aureus. Again, italicize or underline. You don't have to keep saying Staphylococcus, okay? So again, you're just capitalizing the first letter of the genus, putting a period, and then you still spell out the species name correctly. Okay. I am going to go ahead and include the next slide, the apply what you know, in this video, because it is going to kind of be a summary for the types of microbes. So what I'm going to recommend you do is pause this video because I'm going to give you the answers, and go ahead and try and answer these questions just from what you remember. Then go back and look at the previous slide where I described all the microbes and fill this in, okay? So I'm going to go ahead and go through it now. So number one, I am a unicellular prokaryote. Even though I'm often found in extreme environments, I am no threat to you as I do not cause disease in humans. I have a cell wall, but it lacks peptidoglycan. What am I? Well, there's a lot of information here, okay? But notice prokaryote. We talked about two groups that are prokaryotes, bacteria and archaea. So you can automatically go that, narrow down to those two. Both of them are unicellular. So what we need to now look at is the next sentence. Which of those two groups are found in extreme environments? That is going to be the archaea. And because humans don't live and thrive in extreme environments, that's why they are not a concern for us. They're not pathogenic for us. All right, number two, people often mistake me for a plant because my cell wall contains cellulose and I can do photosynthesis. What am I? So by knowing what the cell wall is made out of is normally all you need to identify the type of microbe. The only one we talk about that has cellulose in the cell wall is going to be algae. Remember, they also do kind of resemble plants, but they don't have the root system, so they are not actually a plant, but they do look like. Number three, I am a unicellular eukaryote. So we can rule out bacteria and archaea based on the fact that it's a eukaryote. I come in a variety of shapes and I move around using pseudopods. I do not have a cell wall. So the moving around is telling you that they're very motile. There's a lot of motility here. They don't have a cell wall. Well, we only talked about one organism that doesn't have a cell wall, and that's going to be our protozoans. Okay. All right, number four, I am not made up of cells. That means they're acellular, and I'm an obligate intracellular parasite. What am I? So remember, there are two groups of microbes that are not made up of cells. That's viruses and prions, so we can narrow it down to there. The obligate intracellular parasite is going to be a virus because remember, it's completely dependent upon a living host in order to do anything. All right, number five. My morphology is bacilli. I divide by binary fission and am motile courtesy of flagella. What am I? So morphology is the science word for shape. Okay, so my shape is bacilli, which is plural for bacillus. So anytime you see bacillus or coccus or spirillum, it's automatically bacteria. We only use coccus, bacillus, and spirillum to describe bacterial shapes or morphologies. And then binary fission, that is the process that bacteria use to replicate. So with those two pieces of information alone, we know that this is bacteria. Okay, we haven't talked about flagella or anything like that, but protozoans are not the only organisms that move around, right? And so bacteria are also able to move around. And a lot of these others can too. So we'll talk more about that in future classes. But for now, that is all for this video. Please let me know if you have any questions.